Here is serum albumin. Serum albumin is a protein, in fact it's the most commonly occurring protein in blood plasma. The function of serum albumin is to regulate the blood pressure so that the blood does not leak out into surrounding tissue. Without serum albumin, animals with high blood pressure would suffer huge blood loss. The other function of serum albumin is to transport hydrophobic chemicals around through the blood. These chemicals bind within alpha helix bundles found within the protein. There are about 6 alpha helix bundles within this protein, and each one contains an internal area for ligands. The shape of the serum albumin is approximately spherical, and hence it's called a globular protein. It also has a heart shape, which is appropriate because just as your heart contains blood, so your blood contains many hearts or many heart-shaped proteins. Now, the nucleotide sequence for the amino acid sequence for serum albumin is shown here. Serum albumin consists of 585 amino acids. One of the interesting characteristics of serum albumin is that it contains a large number of disulfide bridges. These are bridges between cysteine residues. Altogether there are 34 cysteine residues in this sequence. 35 cysteine residues in this sequence, and they form 17 disulfide bridges. Looking at the amino acid sequence, there does not seem to be any apparent pattern. However, I decided to see what would happen if we followed each cysteine with a line break. So I created this little bit of software, and when we click on C break, we now see a pattern. As you can see, following each cysteine with a line break generates 3 lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C, followed by 3 more lines, followed by a single C. This pattern persists throughout the protein, and each line ends with a letter C. So there seems to be a regular pattern of 3 lines followed by a single C. In the software, I also counted the number of amino acids in each line, and you can see the length of each line listed down this side here. Now, if we look at these numbers, for example, 10, 23, 44, then if we go down 1, 2, 3 groups, down to this group here, we see the numbers here are 10, 27, 44, which is quite similar to 10, 23, 44. And if we go 3 more groups down, 1, 2, 3, we can see the length of these lines are 10, 27, 44, which is identical to this one, 10, 27, 44. So it seems like there is a repeating cycle every 3 sections, and each section consists of 3 lines. Let's take another one. In this section we have line total 8, 23, 45. When we move down 3 sections, 1, 2, 3, we come to this one, which has line totals 8, 23, 45. So we have a repeating cycle here too. So serum albumin has 30 alpha helixes. These alpha helixes are found equally distributed amongst 3 homologous subdomains. Each of these subdomains contains 3 times 3 lines, and these lines group together into 3 sections. So you could say that serum albumin is characterized by the number 3. In fact, the 27 lines ending in C are 3 times 3 times 3, which group together into 9 groups, partitioned, each one partitioned by a double C, which is 3 times 3, and these form 3 cycles. 3 cycles, each cycle consisting of a repetition of 3 sections in sequence. And these 3 cycles altogether form the total of the serum albumin, which has 30 or 30 alpha helices. So the structure of serum albumin consists of alpha helices and loops, but no beta sheets. The alpha helices cluster into bundles of about 3. For example, up, down, up, followed by a long loop. Now the next bundle is perpendicular to the plane of these helices. So if we rotate this through 90 degrees, there you go. So we're now looking directly down through the original bundle, and you can see in between the helices in this bundle, you can see the ligands binding between them. These are hydrophobic ligands, which include quite a few different substances carried by the body around in the bloodstream. The next cluster has up, down, up, and if we rotate through 90 degrees again until we're looking down through the yellow bundle, so rotate that through 90 degrees, We're now looking directly down through the yellow bundle, and you can see in the middle the ligands binding there too. The next bundle is up, down, up, and then a long looping line. So we can now rotate that one through 90 degrees to look down through that. There you go, we're now looking directly down through this bundle, and you can actually see ligands binding between the helices here too. And the next helices is this one, which is a long loop going from here, and then it goes up, down, and up to here. So if we rotate that through 90 degrees, so we're looking down through those three, again, as you can see inside, you have ligands in this plane too. And now the next bundle is up, down, up. We rotate that through 90 degrees. And you can see the ligands binding between the alpha helices there too. And then the final bundle is up, down, and up. So it's basically up, down, and up. And if we rotate that through 90 degrees, so we're looking down through the bundle, you can see inside there we have ligands binding too. So the finding of this research is that the planes within serum albumin are arranged perpendicular to one another, so adjacent planes of alpha helices bundles are perpendicular to one another. This results in a molecule or a protein which is more open to ligands coming from any direction, since the barrels all point in different directions. Sorry, the clusters all point in different directions. This results in an overall shape of the cluster of a sphericalness to it, hence it is a globular protein. I would hypothesize from this that in general globular proteins consist of subdomains that are rotated at 90 degrees to one another.